Hi there! I'm the Golgi Apparatus. I'm found in most eukaryotic cells and most frequently near heavy protein concentrations. My function is to first receive molecules sent by the endoplasmic reticulum. I modify those molecules and package them into complex molecules. Some are stored, but most are launched and delivered to various destinations within the cell or secreted out of the cell. But I bet you'd like me to clarify this, so I brought in a special guest, Mr. Post Office.
Hello, I'm the post office. I work a lot like my friend the Golgi Apparatus. Just like the Golgi Apparatus, when people come in with their mail, we receive it, we stamp it, and we ship it off to another place in the world.
Vesicles are sent from the endoplasmic reticulum to me, the Golgi Apparatus, where we fuse. I then accept the proteins they empty. I modify them and transport them to their final destinations.
Just like how I fuse all the mail together, then ship them off to their final destination, and also modify them by stamping them as they pass by.
To help you remember, we've made a little animation for you. Proteins in purple are being synthesized inside the smooth endoplasmic reticulum in pink. Transition vesicles in pink, carrying integral membrane proteins, soluble proteins awaiting processing. Processing enzymes and biomolecules pinch off from the surface of the smooth endoplasmic reticulum and are launched toward the Golgi apparatus in pink. Proteins in purple and the transport vesicles are being transported to the Golgi apparatus. The protein transport vesicles are fusing with the cis Golgi in the lower compartment. They have completely fused into the cis Golgi apparatus compartment. As you can see, the Golgi apparatus looks like a pancake-like set of flattened compartments. They are membrane-bounded and are mainly devoted to processing what was carried inside these transition vesicles. Modification begins in the cis Golgi compartment. Steps of modification take place as shuttle vesicles carry the proteins through the flattened packaging compartments, starting from the cis compartment, then going to the medial compartment, and then going to the trans Golgi compartment. One example is a modification of glycoproteins, where sugar are added to the protein through a modification known as glycosylation. When shuttle vesicles reach the last compartment, the trans Golgi, these packaged macromolecules pinch off and are delivered to various organelles within the cell or secreted from the cell. In the animation, one macromolecule is existing through the cell membrane while another macromolecule is being delivered to a lysosome in orange. This completes its outbound path. Golgi apparatus also takes part in an inbound path, which happens at the same time as its outbound path. Inbound vesicles of enzymes and nutrients in blue enter the cell through endocytosis. In the Golgi apparatus, the vesicles are separated and the enzymes are coated with protein to prepare them for transport. Then, the transport begins as the vesicles butt inward and pinch off. Then the enzymes enter the cytoplasm and go back to the endoplasmic reticulum. The Golgi apparatus is a structure with sacs of flattened sacs of unit membranes. Its main purpose is to receive, modify, and package molecules into complex molecules that are sent off and delivered within the cell or secreted out of the cell. These pictures show how similar the post office is to the Golgi apparatus. Mail is received. Then, mail is processed and modified through stamping and weighing. Mail is also processed further by sorting and shuttling. Mail is delivered to specific destinations.